So on 1028, we got yesterday to fertilization, aka conception, and the developmental process. So we've talked a lot about up to this point about sperm, egg development, how they get where they're going, and we haven't talked a lot about conception and development of the embryo and fetus. So that's where we need to go next. Okay. So. Acrosomal reaction. Actually, you know what? Let's come back to this. So 1030, 1031, those should look familiar. I think those are pictures from that video that we watched. On 1037, at the top of 1037, who do we have there? That is a what?
Embryo.
Of what organism?
Human.
What a 7-week-old human embryo there. What do we have at the bottom left?
Is that an animal?
It's a chicken, good. Now, can you tell a lot of difference between the two? No, I mean, if you had looked at those two and didn't know which one you were looking at, you probably couldn't. I mean, I would maybe even think the one on bottom might become human. The one on top looks like an alien still. Not much you can derive from that. So early on in development, almost all vertebrates are very similar in their development. Up until, I don't know, maybe this is 7 weeks, so maybe 10 or 12 weeks, you probably start getting more human features that you can notice. But up until that point, a fish, an amphibian, a reptile, a bird, any other mammal, all those are going to look very similar. Okay. All right. 1039, sperm. So let's say that you are working in a fertility clinic, Mariah, and you've got some eggs that someone's donated, and you've got sperm, and you are tasked with creating, an embryo or embryos for implantation because of, some, I don't know, difficulty or infertility issue. Maybe they needed a surrogate mother for the baby to grow in, and so you have a bunch of sperm you put in with the egg. And all the sperm surround the egg, and all the sperm are trying to get in the egg. Let's say hundreds of them. What prevents more than one from getting in there, Tay? Wrong answer. Let's go here. Number one, Tay, read that for us.
For acrosome?
Yeah, at the top of 1039. Let's just go through the picture in the book here.
Contact. The sperm contacts the egg's jelly coat, triggering exocytosis of the sperm acrosome.
Okay, the acrosome, remember, is the part on the outside with the digestive enzymes. Exocytosis, what does that mean?
That's like something's coming out of the cell.
Exiting the cell. Okay, so the acrosome is exiting the cell, and it meets that jelly-like coat on the outside of the egg. Number two, Walker.
Acrosomal reaction. Hydrolytic enzymes released from the acrosome make a hole in the jelly coat.
All right, hold on a second. What is a hydrolytic enzyme? It's an enzyme that uses what? Process.
Hydrolysis.
Hydrolysis. What is hydrolysis? Hydrolysis. What?
Splits water. Or uses water to split something.
It uses water to split stuff. Good. So hydrolysis is the breakdown of stuff using that water. Remember, the H and the OH split off. So it's using water to break something down there. Okay, keep going. Growing.
Okay, growing actin filaments form the acrosomal process, which protrudes from the sperm head and penetrates the jelly coat. Proteins on the surface of the acrosomal process bind to receptors in the egg plasma membrane.
Okay, so in this case, the sperm is acting like a what? Nope. What? Ligand. You see how it's attaching with the receptor proteins on the outside of the egg? So that sperm is acting like a ligand in that case. It's attaching, it's a receptor, it's a signal molecule that's attaching to those receptor proteins on the outside of the egg. See, they're all over the outside of the egg, so the egg's just waiting. It's not like you got to hit one spot if you're a sperm. They're all over the outside of the egg. It's trying to attach to those to signal to the egg. First one here. It's kind of like running through the ribbon. You the first one there. Okay. Number 3, Sarah.
Contact and fusion of sperm and egg membranes. Fusion triggers depolarization of the membrane, which acts as a fast block to polyspermy.
Fusion and depolarization. So we have a signal that's being sent throughout the egg, okay? And let's see, depolarization of the membrane. Okay, so you'll notice here, this term sounds like something I would make up off the cuff. A fast way to block polyspermy. Polyspermy would be what? Multiple sperm getting into the egg. Yes, so this is a way of preventing that. Number 4, Matthew.
Cortical reaction. Cortical granules in the egg fuse with the plasma membrane. The secreted contents clip off the sperm binding receptors and cause the fertilization envelope to form. This acts as a slow block to polyspermy.
Okay, so what happens after that one sperm binds? What happens to all the other receptor proteins?
Kind of like come off.
They detach, they fall off, they break loose, yeah. And so in doing so, no other sperm can attach to them and prevents any other sperm from fertilizing the egg, okay? So that is how we prevent polyspermy from taking place. So in your notes, basically we went through this. Acrosome releases digestive enzymes. The acrosomal process eats its way through the jelly. The sperm makes connection with the egg on its receptor hands. Connection of the first sperm causes the receptor hands to be released and prevents multiple sperm from fertilizing. Number 3, sperm begins to enter the egg cell through a hole in the membrane. The DNA in the sperm will determine the organism's sex. So I think y'all probably remember Henry the Eighth. What was his big problem? What did he want?
He couldn't get a son.
He wanted a male heir, and so what did he do with all his queens?
He got rid of them.
Either killed them or got rid of them, okay? What was the problem with that, Nathan?
He still didn't have any kids.
Well, I mean, like, what was problematic other than him, killing people and getting rid of them? What was the irony in all that?
He never was able to have a son.
It was his fault.
It was his fault. Who determines the sex of a child?
The man.
The male. So he was blaming it on the female, his wives, and in reality, he was the one responsible for the sex of the child. The male releases sperm that either have an X chromosome or a Y chromosome. Females, what are their sex chromosomes? They've got 2 X's. What'd you say?
I said all X's.
Yeah, so they've got 2 X's. So girls, in all of your eggs, your sex chromosomes are going to be what?
X's.
You're only going to have an X to give. So it's one X or the other. There is no option for you. You have no other option as far as whatever you're giving. But when it comes to the man, he can either give an X or he can give a Y, and then that's going to determine what the sex of the child is. So he is solely responsible for whatever the sex of the offspring are. Okay? Sperm entering the egg causes a rate of cell respiration and protein synthesis to increase, because what's going to happen as soon as that DNA gets in there and fuses with the egg of the DNA? So sperm and egg DNA, they fuse, forms a zygote. What's going to happen next?
It's going to start dividing.
It's going to start dividing. It's going to have rapid division take place. So you got to have an increase in cell respiration. You got to have a lot of energy for that to take place. You've also got to have a lot of protein structures that are being used proteins and enzymes that are being used in that process. So there's going to be a rapid increase in those 2 things as soon as fertilization takes place. Part C, Miranda.
On the note,
Yes.
egg undergoes a second meiotic division of the mother's DNA into a haploid state. This only happens if the egg is fertilized.
All right, turn back to 1023 real quick. So on 1023, we looked at this the other day. It's one of the stranger things, I think, in terms of egg development. We talked about eggs quite a bit yesterday, and how, you only have so many eggs. You're really only releasing about 400 to 500 in your lifetime, ladies, even though you have 500,000, 1 million, 2 million. I don't know. These people can't count or make up their mind. But you got a lot of them, but only a very few small number relative are released for fertilization. And then, in most lifetimes, if you think about a woman, let's say she has 1,000,000 eggs, really, on average, only 2 of those actually wind up getting fertilized and passed on. So it's a real crapshoot as far as, which one actually gets its DNA out there and what the kid turns out like. So, I think one thing when we think about ourselves coming from eggs, we are very, kind of random and unique in our characteristics because of that.
So do the ovaries produce like hundreds at once, or just like?
Well, you're born with all the ones you're ever going to have.
Oh.
So they're already born with them. And you can see here, they're in this, at the top of 1023, Nathan, where it says the primordial germ cell. The oogonium, this is what they look like to start with, like when they're born. Or when a woman is born, she already has all these. It's not like a man who's producing millions all the time. These you're born with if you're a woman, and then once you hit puberty, then you start to see this meiosis. Sorry, not mitosis. Meiosis takes place where DNA separates out. So you get rid of the pairs of chromosomes. You get it down to 23. And then only if a sperm enters the egg, you can see at the bottom there, does the sister chromatids of the egg split off. So you reduce it down to just 23 different sister chromatids.
So is there like a problem where like the eggs wear down over time since they're there for so long?
Well, that's actually a very good question. So I don't know how we got off on this morning. There's no telling what I said. We started talking about sperm and egg donors. Typically, I'm going to say sperm donors, they're looking for guys that are in their 20s. And egg donors, it's usually the same because sperm donors, I don't really know. Generally, the older you get, the more problems there are, but I don't think it's that big a deal. But with women, it's a really big deal when you're talking about egg donors. Somebody asked me, how do you go through the process of egg donation? Like, they're injecting you with hormones. You're releasing eggs. You may release multiple eggs, and then they go in and collect them. The egg's a pretty big cell. So, it's not ginormous, but it's a pretty big cell. So you could see it with the naked eye, which is one of the few cells you could see with the naked eye. And so they go in, they harvest those, and then they can keep them. I don't know if they freeze them or maybe they freeze them or suspend them, put them in a suspended state for a while. A lot of women, there was a news article like in tech companies out west, they were paying for women that worked there to freeze their eggs. So women were getting like their late 30s. They're like, I want to have a kid. And the tech company's like, well, we'll pay for you to freeze your eggs if you want to hold off on doing that so you can keep working. And so it's a very expensive process, but it's one of those things. I'll be honest with y'all. We had kids when I was, 30 and 32, and I cannot imagine being 40 and having babies or later and having babies just because I feel old and tired. I have to get up with my dog sometimes to let her out in the middle of the night because she eats a bunch of pears and has diarrhea all night. God bless America. So if I don't let her out, she poops all over the house. Anyway, it's kind of like having a kid, but I cannot imagine having kids. I cannot wait till all the pears fall off the tree. What were we talking about? Eggs. So donate. Back to your question. Eggs. The older a woman gets, so when you're talking about Down syndrome, there's other disorders, but Down syndrome is probably the most notable because a lot of kids are born with it. If a woman, when she gets to about 35 and then to 40, just imagine there's a graph of women and how they have kids with Down syndrome. It's almost nothing, like 1 in 1,000 and then 1 in about 700. And then once they hit 35 and get up to 40, it basically goes like that. It goes into being like, once you're 35, 40, it goes into about like 1 in 100, 1 in 70 or 80, I think, once you get to 40. So it's a much higher number when you get later on. And the only explanation I could think of is that second division isn't complete. And so there's also a lot of, Down syndrome is when the 21st chromosome, one of them doesn't separate. There's also a lot of like trisomy 1 and 2 and 3 and 4 and 5. You just hear about Down syndrome because
It's the most common.
I actually don't know if it's the most common. You have to look that one up. It's the most common one you see in, I guess, people that are living, because the 21st chromosome is like the smallest one. It contains by far the least amount of DNA, so it affects the least amount of stuff. So if you're talking about like trisomy 1 through 10, you're talking about some major developmental disorders that are taking place there. And speak up, don't forget about your developmental disorders. So a lot of those kids aren't going to make it probably past the first trimester. They're probably going to be resulting, what do you call it, a miscarriage, because just development's not going to take place like it should. Yeah.
Are we ever gonna learn about like each chromosome in here?
Each one, what they have on it? Yeah, we don't have that time. We're mostly going to group we'll lump 1 through 22 together as the non-sex chromosomes, the autosomes, and then we'll talk about 23rd pair, which is the sex ones a little bit. But yeah, we don't have time for that. There's websites though where you can go and look at like each chromosome, and you can kind of skip down through there, and it will tell you genes for different stuff that you find on them, like hair and eye color and skin color and stuff like that, and like where different cancer genes are found. So there's websites that have, a lot of genes mapped out and what chromosomes they're on. So anyway, yeah, we don't have time for that though. Let's skip down to the egg DNA and the sperm DNA fuse together to create the diploid state, the zygote. The tail of the sperm. Okay, so the sperm isn't completely useless, other than the DNA it has. It does a little bit. The tail of the sperm is broken down to help create spindle fibers for mitosis. Good job. And the basal body of the sperm detaches to help create centrioles. So it does contribute what it can. But like I said, the sperm, there isn't much to it. All right. Now, let's look.
So,
1028, yes.
with the sperm and meiosis, so the first division is like replication of all 46.
So the first division meiosis is going to be, so you've got 46 chromosomes. Each one has a sister chromatid. You're separating it down to 23 pairs.
Okay.
So it's reducing the chromosomal number. But each one of those 23 that you're left with then has a sister chromatid.
Oh, okay.
Yeah. And then the second one is the sister chromatid separating, so you still wind up with
and
4 cells.
in eggs, it's just making a copy and letting it go, like the first polar and second polar bodies and stuff.
The first polar body is getting rid of those 23 chromosomes, and then the second polar body is getting rid of the 23 sister chromatids.
Okay.
If fertilization takes place, yeah. Like I said, I think it's kind of weird that it waits, but who knows? And like you said, I think it's a good point. I think that's probably why you see a lot of chromosomal disorders and why there's they always reference the mother and her age. So I would suspect that, yes, the older it is, that has something to do with the inability or improper separation that takes place. Okay, where did we go? Let's go to 1028. Number 1, the egg's released. Number 2, fertilization occurs. Number 3, cleavage starts to happen. So the cell starts to divide. Excuse me. Number 4, cleavage continues. You see this ball of cells. And then hopefully by about day 7 on number 5 there, it makes its way down and implants inside the uterus somewhere. Okay? So now keep in mind, I'm going to give you all a lesson here when you go to the lady doctor one day and you're trying to get pregnant or you're pregnant and figuring out what's going on. So the first day, like when you're counting the 40 weeks, the first day of your pregnancy is the last day, or the first day that you started your last period, your last menstrual cycle, okay? And then how long after that is it going to be before conception actually takes place? 2 weeks probably, yeah, a couple weeks, assuming you're on like a normal cycle. And then before you find out, you're already at, so let's say 28 days, you miss your period, you take a pregnancy test, you go get a doctor's appointment, you go in maybe sometime that week, So you're taking a pregnancy test, you go in, they give you another pregnancy test. If it's positive, then you'll go in and get a sonogram. So you're about at 5 weeks at that point, 5 or 6 weeks. You go in, and usually by that point, you're going to see a heartbeat or hear a heartbeat. Really, it's about the size of like a peanut. I always call my daughter Peanut because that's what she looked like the first time I saw her. So, I mean, it's super duper small. But it already at that point, after conception, shortly after conception, you've got blood, you've got organs that are forming, you've got a heart that's beating that you can hear rapidly thumping. So development's taking place super duper fast there at that point. Okay. And then I'm gonna, let's see here. Where do we go to? Cleavage. Cleavage. Rapid division occurs to create a morula, a ball of stem cells. We're going to come back to this baby in utero. We'll talk about that later. And that baby. All right, let's flip back to the other page. Turn it over to 1041. So on 1041, it's funny to think we all started out this way. We think we're all big and bad and well developed, which we are. But we all started out as one cell, okay? And then the cell divided in 2, and then to 4, and then to 8, and then 16, and so forth. But it doesn't just keep a solid ball of cells within that egg that it originally started, okay? It does for a while, and that's called the morula. So that's important. Those cells initially early on in that morula stage, those are what type of cells? It's in the notes. I just read it. Stem cells. What are stem cells, Walker?
Are they like body cells?
They could be.
Don't they say they can become any type of cell?
Stem cells are cells that become any type of cell, right, Clive? So, they are said to be, I'm going to write this down, you'll see it later on. They are said to be totipotent, meaning like they have a total potential to become any type of cell in your body. That's why there's a lot of research. Where's my chapstick? There's a lot of research into stem cells because if you had like a bad liver or bad heart tissue or kidneys or something like that, that there was something wrong with them, well, if you could take stem cells, generate new tissue and implant it, then it could function just like yours was supposed to. And it would have your DNA in it. So your body you wouldn't have to worry about rejecting it. So that's why there's so much research that goes into stem cells. Early on, they're totipotent. Then at some point, as they're dividing, let's see here. Cells get smaller and smaller with each division. Remember, the egg's super big. The cells aren't going to grow there early on. It's not getting any bigger like around. As it divides, the cells just kind of get smaller and smaller within that original shape. Okay? Cells do not go through a normal G1 or G2 phase, only S phase and mitosis. So they're not having to grow, so division can take place really quickly early on. The morula continues to divide but hollows out to form a blastula by blastulation. So I'd probably underline that, the blastula part. This is a hollow ball of cells. Why does it want to be hollow? I'm sure there's a fluid in there, but they're not cells. Why is it hollow on the inside? You're getting there. What? It's not actual tissue in there. That's going to be the digestive tract, kind of. So it's going to give it somewhere for it to fold in and form that tract that goes from one end to the other. If you think about all animals from worms all the way to us, you're pretty much excluding jellyfish and sea anemones and some like sponges. They don't have a tract that goes from one end to the other. But pretty much all other animals, you have a mouth and an anus where you have a tract that goes from one end to the other and digestion occurs in the middle. And so this hollowness on the inside helps to fold in and form that, okay? So, the blastula has 2 layers of tissues now. Now flip over to, you can see that at the bottom of 1042. It's divided, it started to form that blastula. Then turn over to 1044. So that's going to start to fold in on that hollow ball of cells. The blastula has different regions with different size cells in it. Read A, B, and C for us, Matthew. in the notes? Get your life together. Print those off. Walker.
A, small cells on top, animal pole makes the head. Large cells on the bottom, vegetal pole makes the bodies, and the hollow space referred as the blastocell.
Yeah, I'm not too worried about those. The key part here is understanding that you're going from a round ball of cells, and then you've got to start elongating at some point. We don't want somebody that turns out to be the blob, right? So you've got to have cells that are kind of migrating to different ends and that are going to separate and spread out. Eye part, blastula. Read down for us, Matthew.
Blastula continues to develop into three tissue structured cells called a gastrula and bigastrulation.
All right, gastra. When you hear that term gast, what is that referring to? Like gastric bypass. What?
Like gases.
What about in your body?
It's excreting out.
Your digestive system, good. Somewhere in there. Like gastric bypass is like when your stomach gets, they shrink it or cut it down. Anyway, so this is talking about, it develops into three tissue layers called a gastrula. So this is important too. The third layer is created by an involution. A cell's moving inward from the outer surface through a hollow hole called the blastopore. You can see this at the bottom of 1044, this blastopore. In humans, this blastopore is going to form our what? Here's where it gets interesting. Yes, this is where it forms your seventh planet. Very good. Okay. And that's how all vertebrates work and all echinoderms, things like sea stars, sea urchins, what do you call those things we find at the beach? Sand dollars, yeah, all those. So all vertebrates and all these weird grouping of non-brain things like sea stars and sea urchins, they all form this way, where the blastoform, when it folds in, that forms the anus first. These organisms are referred to as deuterostomes. Okay, that's important. A lot of times that comes up on the test. So as this tube starts to form, which is going to be part of your digestive tract, the anus forms first. Okay. Now, not all organisms form that way. Protostomes form the mouth first. So when the blastopore folds in, it's actually their mouth that forms first. These are your worms, arthropods, mollusks. So those organisms form the mouth first, and they're called protostomes. I don't know why one does one and one the other, but whatever cranks your tractor, as my grandmother would say.
Does this come up on tests together, or is it usually just...
Yeah, usually they come up. Well, I don't know. You might see one or the other. Remember, we're deuterostomes, and that is anus first. I don't know. I mean, maybe the easy way to remember it, I always associate alphabetical stuff. Those like anus and deuterostome. A and D is closer in the alphabet, and M and P are closer. So whatever it takes for you to remember stuff, that might help out. All right, so in your book, you can see here there's three distinct tissue layers that are starting to form from this little round ball of cells. At the top of 1045 here, Sarah, you can see we've got the ectoderm, which your note writer kind of simplified this for you, which is probably all you really need to be familiar with. But the ectoderm makes the skin and the CNS, which is our, central nervous system. If you look on the book on 1045, it goes a little more in detail. The outside of the skin is really what's forming here. Nervous and sensory systems, pituitary gland, adrenal medulla, jaws and teeth, and germ cells. So all of those are forming from that outer layer of cells. Okay? Ecto means?
Going in?
Ecto means outside. And so we'll get to the other one in a second. Mesoderm. Meso means middle. So the mesoderm, this is where, and I'm just going through the book one because it's so much more detailed. Skeletal, the muscular systems. So this middle layer of cells starts to form skeletal, muscular, circulatory, which is your Circulatory system is your blood, heart, lymphatic systems, excretory, reproductive systems, except for germ cells. The underlying layer of skin called the dermis, which is under the outside, and then the adrenal cortex, which is on the outside of the adrenal gland, which seems very strange. So that is formed from those mesoderm cells. And then the innermost lining of cells here, the ones that are yellow in your book here on the right side, is the endoderm. Endo means inside. So there's some big words here. I'm going to simplify these real quick, make them easier for you here. Y'all haven't taken anatomy, and y'all shouldn't really be familiar with these. The endothelial lining of digestive tract, endothelial line of respiratory, excretory, and reproductive tracts and ducts. When it says epithelial lining, that's talking about basically, that pretty much includes any orifice that you have on your body, your like mouth, and goes into your stomach or your respiratory system or into your reproductive tract. It's the lining on the inside of that that's exposed to the outside. So any lining that something could come in from the outside, that's made from that endodermic tissue, okay? And so those cells, Mason, are said to be, they're not totipotent because they can't become anything. They're said to be pluripotent, Pluri potent. Pluripotent means they can't become anything, but they can become multiple things. So they can become multiple different types of tissue in the endoderm or in the ectoderm, okay? And then they continue to develop from there. The blastopore fills in after gastrulation with a yolk plug. I'm not too worried about that either. Gastrulation, hopefully a 25% chance of implant in the uterus and development into an embryo. Embryo is that developing organism early on. I'm going to say, I don't know for sure numbers, but implantation is really going to depend on the person. Like, different women have different environments for babies to grow. Y'all probably heard about people who, and guys, y'all probably don't hear these conversations. Y'all probably watching football at the dinner table. But if you've got a friend or family member who's having difficulty getting pregnant, sometimes you might hear somebody talk about, they've been trying and they can't get pregnant, or they've had a bunch of miscarriages. Sometimes, it's something maybe in the endothelial lining of the uterus or whatever, but sometimes it just takes people a long time to get pregnant, depending on what the environment and how the baby grows and implants. Some people, they could get pregnant the first month that they start trying. The gyno, my wife's gyno, Dr. Ray, y'all may know her. She said when we went in, I think she said usually it takes 6 to 9 months, or maybe a little bit longer, on average for most people to get pregnant when they start trying. So it's not as quick as you think a lot of times, depending on what's going on and who the individual is. And you can see here with all this development and stuff that's taking place, all this folding and forming and becoming new tissues, there's a lot of room for stuff to go wrong here, early on especially, okay? That's usually why most people recommend you don't tell anybody you're pregnant until you're at least 8 weeks. A lot of people that have had trouble or have had miscarriages, they won't tell people until like 12 weeks a lot of times when you're almost through with the first trimester. Because usually that's kind of a milestone. Once you make it to that point, everything looks good on the ultrasound, then there's a good chance that things are going to be, okay. Although things can happen later on. Placenta. Organogenesis, sorry. Flip over to the next page. 1047, you can see kind of this implantation process and development into the 3 tissue layers. On 1049 here, you see this, what is this thing? A chicken embryo. Could have been us for all I know. You can see how the eyes and the heart and blood vessels and stuff have started to form there. So that's really the early stages of organogenesis, where the organs start to develop. If everything's not right at that point, then you usually have major issues with an organism and its ability to survive. like a human. Like they will go in, one of the things they do on those early ultrasounds is they're checking really all the ultrasounds once they get big enough where they can see. They're looking for like, is the heart developed? Is the lungs developed? Lungs is usually last. Is the liver developed? Are the kidneys developed? Those are all things that they're checking and looking for in those ultrasounds because they're things that they want to be aware of, that they want to make the parents aware of. So organogenesis, the most important time is early on. All right, turn back to 1029. I know I'm throwing a lot at you. All right. Been talking too much today. So on 1029, we got a baby in utero here. All you Nirvana fans. The placenta. The placenta is a what? It is a dish-shaped organ containing both embryonic and maternal blood vessels. So this is technically an organ that grows and forms during childbirth. That might be on the test. Who knows? This is what connects the mother and child. Develops from cells at the bottom of the gastrula. How do we connect? How do we know we connected to our mother? What evidence do we have? Your belly button, right? So you got a belly button. You probably don't have any umbilical cord left. Let's hope. But that belly button shows that you were connected to your mother at some point. Good. And that's where we are connected with them. Mother's blood vessels and fetal blood vessels do not mix. There's like a separation you see here. And basically what's happening is a diffusion of nutrients. So kind of like at the blood-brain barrier in your brain where blood doesn't go into where the brain is and you have cerebrospinal fluid, you're diffusing nutrients and waste products back and forth across this barrier. Let's see if we can read a little bit more of that. Mariah, read figure 4616 for us.
From the fourth week of development until birth, the placenta, a combination of maternal and embryonic tissues, transports nutrients, respiratory gases, and waste between the embryo or fetus and the mother. Maternal blood enters the placenta and arteries, flows through blood pools in the endometrium, and leaves via veins. Embryonic or fetal blood, which remains in vessels, enters the placenta through arteries and passes through capillaries and finger-like chronic villi, where oxygen and nutrients are acquired. Fetal blood leaves the placenta through veins leading back to the fetus. Materials are exchanged by diffusion, active transport, and selective absorption between the fetal capillary bed and the maternal blood pools.
Okay. Yeah, that's about all you need to know. So, the placenta is very important in exchange of nutrients. Now, in your notes there, hormones such as progesterone and HCG are helping the development and communication with the mother's body. The human chorionic gonadotropin, I know it's hard, we'll just say HCG because that's hard to say. That is what they look for in pregnancy tests. So pregnancy tests, they're looking for that HCG, and you can, sometimes before even a missed menstrual cycle, you can test for that and find out if you're pregnant or not. So they're getting better and better at that. Development of the amniotic sac occurs, contains salt water, same concentration as ocean water. The kid can pee in there. I used to have a friend who was a med student and said it was mostly pee later on, but I'm not sure how much. They are not supposed to poop in there because of exchange. It would be very toxic, harmful environment. Kids can poop early on, like during labor if it runs too long, and like it can make them sick in there if it gets in there and they're swimming around in it. So there's a name for it. I can't think of what it is. But yeah. Now, two things you need, or something you need to be familiar with here. Development of the amniotic sac and the amnion can also be found in birds, reptiles, and monotrim eggs. This is an evolutionary question. Organisms that either have an amniotic sac or an amniotic egg, they can be born where?
In harsh environments.
More specific.
Salt.
What?
I don't know.
What do you mean harsh environments?
Well, I mean, if it's, I don't know.
Where can mammals, birds, reptiles be born that fish and amphibians cannot be born? On land. So the amniotic egg is a solid structure that helps them prevent from drying out that you do not find in amphibians or fish. And the amniotic sac is an internalized version of that where it allows individuals to keep their babies and be born away from land and further develop. Okay? Monotremes, FYI, in case you didn't know, these are your egg-laying mammals like the duck-billed platypus and the spiny echidna. They are still mammals. They give milk. I don't think they have nipples. I think the echidna, I saw this guy one time. They had laid an egg. It looks like a little bird egg. And so they had the baby, and she started producing milk. And just think of like the hairiest chested man you could think of. And he kind of squeezed her chest after she had these, and you could see the milk ooze out of it. And so the baby kind of like hatches out of this little egg, climbs up on his mama's chest, and then like hangs out there and licks milk off her hairy chest, I guess. Can't believe I'm putting this on the internet. Anyway, so it's just a very strange. It could be why there's only like a few different types of egg-laying mammals, and then everybody else went on to, doing other stuff, like having a placenta and or a pouch, which made a little bit more sense. But anyway, let's see. Second trimester. So the trimesters are basically broken up into 13 weeks, and she got 40 weeks total. Second trimester, the human's already pretty well developed, growth and size, and then the third trimester packs on a lot of weight. The last month or so, I think you're putting on about a pound a week. Maybe it's the last 2 months you put on a pound every 2 weeks. Anyway, it's where most of your weight gain occurs. Because if you're only at 7 months, at 28 weeks, you're probably only going to be a few pounds. I had a friend who was, I think, 24 or 25 weeks. She used to go to church with us, and she was born, I think she was like 1.3 pounds or something like that. Very, very small. Now, what's the biggest issue with being born early with humans? And I imagine most other animals. What develops last? Brain's pretty early on. This girl was like a nurse practitioner. Her brain was still there. What do you think is going to develop last? Respiratory system. Good. So that's usually the biggest issue. They have to make sure they're in those little NICU, little incubators, and getting plenty of oxygen. And so that's usually the biggest deal for those early term babies being born. So birth, how does it happen? Oxytocin, we already talked about this. It causes the uterus to contract, and we talked about that positive feedback loop. Lactation is also induced through prolactin, that hormone. Convergent extension. This is going from a round structure to elongated. Nathan, read A and B for us.
Gastrula round going to the embryo elongated structure.
Okay, so we've got to elongate, and this happens pretty early on, about the second week after conception. C, Tay. B, Tay. I can't even make up my mind here.
The cells move using the extracellular matrix into a single file line, causing the structure to go from round to elongated.
Okay, so those cells have to migrate into that separating out or elongated structure. Protein receptor hands are communicating between cells. You don't really need to know the CAM part there, but those receptors are communicating to make sure they separate out and they get in the proper position. And then you have these apical ectodermal ridges, which are on the outside, responsible for making your arms, your legs, or wings, or whatever type of organism. That's not really important either. So, any questions? I will say this on 1031. You guys saw the birth the other day when the child came out. What did you not see being born? This is what they don't tell you, Tay. What?
I didn't see the placenta.
The afterbirth. So usually they'll cut and snip off the umbilical cord to make sure to stop any bleeding. And eventually what happens is the placenta detaches over about 30 minutes. So ladies, not only do you get to have a baby, about 20, 30 minutes later, that you have to deliver all this afterbirth, the amniotic sac and the placenta and umbilical cord, all that has to come out. And I remember my first kid, they threw that placenta up on a tray And I swear it looked like an 8-ounce steak that had been put in a blender for about 3 seconds. And I thought, I could see how people might eat this in some places, but it wasn't for me, Matthew. Y'all have a good lunch.